Now, our first keynote speaker is Anya Plutynski. Anya is an associate professor of philosophy at the Department of Philosophy of the Washington University in St. Louis. In her work, she has focused predominantly on the topics of evolution and cancer. This includes her research on the history of the Cancer Genome Atlas project and the topic of precision oncology. In 2018, Oxford University Press published her book explaining cancer, finding order in disorder. And if you may be so kind and share your presentation, you may start as soon as you feel like. And please unmute yourself also.
Okay, thank you so much, Martin. I'm thrilled to be here, as it were. I'm really excited to hear from you all. I'm really sorry that we all couldn't be in person and see one another. It's unfortunate. But I want to thank especially Martin for organizing this. I think it will be a fantastic group of speakers, and I really am looking forward to hearing from all of you. I also, before I get started, because I'm not going to be able to cite everyone whose work I'm citing and whose work influenced me, I just want to mention a few of these people. Several philosophers have been writing about big data and big data science, particularly in context of medicine, and I want to just mention a few of these people whose work has been really influential in my thinking about these topics. So I'm probably going to say things that many of them are familiar with and know quite well. And then some of the scientists I interviewed in service of this project. So I've been doing interviews with participants in the Cancer Genome Atlas project and asking them about their research and what motivated them and what challenges they faced along the way. And so these are some of the people that I've interviewed so far, and a lot of their ideas are very much present here. So I wanted to thank all of them as well. So all of you will be familiar with this image, this sort of iconic image by now of our sadly former President Obama announcing the Precision Medicine Initiative in 2015. And with this initiative, he funded what became the All of Us project, which is a major effort at sequencing 1 million Americans and following their health data. But he also spoke of ramping up cancer genomics. So he devoted a significant portion of those funds to research into cancer, and in particular, precision medicine in cancer. So I'm going to talk today about precision oncology, sort of the state of the art and where we are and where we've come from, and sort of try to contextualize it and speak to some of the major challenges facing precision oncology, as well as the hopefulness of applying machine learning in this context and the challenges facing applying machine learning in this context. So perhaps ironically in 2015, actually, the Cancer Genome Atlas project was winding down. So it's kind of interesting that he spoke of ramping up cancer genomics, because this was actually the end of a 10-year project starting in 2005, aimed at mapping the genomes of 33 major cancers. And the goal, as sort of described in the program statement, was to identify the changes in each cancer's complete set of DNA, its genome, and understand how such changes interact to drive disease, to lay the foundation for improving cancer prevention, early detection, and treatment. So this has been a project that was sort of very much in the mind of Francis Collins after the completion of the Human Genome Project, and it was very much modeled on the Human Genome Project. They followed some of the same strategies of multi-institutional research and putting these institutional research programs into competition with each other for both speed and efficiency. And the participants I spoke to thought of it a lot like the Human Genome Project, as a sort of division of labor. Different institutions were involved in different parts of the project. And the goal ultimately was really, I mean, here it sort of is the program statement, but I think the goal was as much to improve the speed and efficiency of both the technology and the cost, and to improve the sort of institutional structures around cancer genomics in service of these downstream ends. So these were the 33 major cancers sequenced, and what they did is they collected samples, interestingly enough, quite different numbers of samples across these different cancers. So here, 1098 breast cancer patients, 470 melanoma patients. The goal initially was 500 each, but they didn't quite meet that, particularly for the cancers that are relatively rare. So they took samples or biopsies of both the tumor tissue and then the healthy tissue. They did what's called mutation calling. So they identified variants in the cancer tissue that were distinctive to that tissue. And then they gathered all that data for each cancer type and subtype to identify what are called driver genes, genes that are taken to be largely responsible for the behavior of cancer cells, whether the capacity to grow without limit, to resist apoptosis, to invade, to metastasize, to break down surrounding tissue. And so all these marker papers, which were the sort of consensus, they call these the consensus sequences of each of these major cancers, were published in journals like Cell and Nature, and sort of set forth as the Cancer Genome Atlas's consensus statement on each of these major cancers. But it was a sort of slow growing process, and the first 5 years, according to the participants I've talked to, were really a learning process. They were sort of getting things off the ground. They weren't entirely sure what they were doing. They were sort of building the ship as they flew it, if you like, or building the plane as they flew it. And what the 2015 funds went into was the sort of downstream next generation project that was sort of well underway actually by 2015, drawing upon the Genome Atlas data to generate sort of further information, both about the sort of digging down deep into the oncogenic processes and better understanding how these operated, but also looking at patterns across these different cancers. So finding common pathways and different pathways across different cancer types and subtypes in service of developing targeted therapies that ideally would be able to effectively intervene on different cancer types. So regardless of cell of origin or tissue of origin, developing these therapies or identifying therapies that could be used across different cancers. And this is just one of over 60 papers that were published as a result of the pan-cancer Atlas project. This was aimed at identifying these patterns. So this is one of several examples of the kind of science where what they're doing is they're sort of it's a drilling down into the data and using both computer algorithms and artificial learning mechanisms to identify patterns and process across vast amounts of data, right? And identify these patterns in service of finding commonalities and differences. And what they found in this case was that the cell of origin determines largely the types of mutations common to a particular cancer, but they also found that in some cancers there was different, that you could find more similarities at the molecular level, even though they were different cells of origin or tissues of origin, which was pretty interesting. So there are a variety of different uses of machine learning in cancer research. I've identified one here. This is the upstream, identifying patterns in genomic data, finding correlations. But there's also more downstream applications, which are linking those variables to health statistics. So one of the big challenges of the Atlas project was they didn't always systematically collect clinical data. They collected some clinical data, but it wasn't always as systematic as they hoped. So now they're going back into this and mining it and developing databases that actually link the genomic information with information about downstream outcomes and treatments, and say time to recurrence after this or that drug administered, They're also looking at it in order to develop diagnoses. So I'm going to talk about one example of mammography where they're trying to identify patterns in mammograms in order to determine which cases are cancers and why or why not. And then developing drug line responses and looking at patterns across this. So massive correlational studies and finding similarities and differences in drug response of different cell types with different mutational profiles. So one of the big pushes behind machine learning in this context is or one of the ways in which it's often framed is that this is theory-free or a hypothesis-free science. It's a new kind of science. It's a radical kind of science. It's going to revolutionize how we approach science. And I think that I'm not the first to say this, but I'll just say it again because I think many people are well aware of this by now. But a lot of the science is not theory-free, and I'm going to talk about why it's not theory-free, that you can't do hypothesis-free science. So I'm going to talk about why it's not theory-free and where the theories are getting in and what kinds of presuppositions are driving a lot of the research. I also think that sort of one of the principles behind it, the idea that if we just gather more data that will remove various sources of bias and thereby sort of reduce the problem of the curse of dimensionality. So one of the sort of ways that this kind of research is promoted is people will say, well, we can use machine learning to sort of reduce the dimensions of the problem. And upstream issues in data quality, I'm going to argue, need to be addressed prior to addressing these curse of dimensionality problems. Another big issue is that sometimes this is framed as a sort of purely genomic issue. And one of the things that all of the downstream research is now looking into is integrating a variety of different sources of data, from upstream genomic to transcriptomic to now methylomics. There's a whole variety of different ways in which different kinds of data at different scales need to be better integrated in order to understand how to develop effective targeted therapies. And last but not least, this could be a talk in itself. There's a variety of critics, particularly oncologists, people who are working in the clinic with patients, who are concerned about overhype and regulatory control of a lot of the development of these drugs and therapies. So there's concerns about financial conflict of interest, inappropriate surrogate endpoints, and claims to benefit that maybe overstretch what we reasonably can expect. And I'm going to talk about some examples of trying to extend some of these therapies beyond what the basic scientists might think would be a reasonable way to go about things. But I'm going to start with the upstream question because one of the things that I've been thinking about a lot is where we got this data from and what it looks like and what's the data quality that we're working with. So I want to talk about some lessons I've learned from talking to people involved in the Cancer Genome Atlas project and how the upstream data just both producing it and analyzing it faces a lot of complex issues that people are still sort of working with and trying to better understand. So this is sometimes called the Cancer Genome Atlas pipeline, and I've described a bit of it already. The idea is that we gather these reference tissue, the healthy tissue, and then the tumor samples. The next stage is the sequencing and mutation calling, identifying the variants distinctive of this or that cancer, and then developing this consensus sequence. And at each stage of this pipeline, there are sources of bias and error. So both with respect to the quality and the quantity of the samples and their representativeness, with respect to the tools and strategies used for mutation calling, how things are ruled out, how things are ruled in, and downstream, of course, in analysis. So I'm going to talk about one particular challenge that a lot of researchers faced when they were developing tools, algorithms to analyze and sort of compile this data into a consensus sequence when they ran into surprises. So this is literary license because normal genomes are not, of course, all alike. But here's Meyerson, one of the major researchers involved in developing these algorithms for analyzing cancer genomes and better understanding cancer genome heterogeneity. But I like it because it's Anna Karenina, so here we go. So he says, normal genomes are all alike, but every cancer genome is abnormal in its own way. So what happens when cancer cells sort of arrive on the scene is they arrive with all sorts of problems with both genome division and development and cell division. And so they acquire a variety of, they're unstable, okay? They have a variety of variations in them, some of which are driving or playing a causal role in the behavior of cancer cells, and many of which are what are called passenger mutations. And they're all quite variable, not only because of the acquisition of these mutations, but also because, of course, they inherit variability from their ancestral genomes. So normal genomes are not, of course, all alike. And right around 2012, about halfway through the Cancer Genome Atlas project, as they were developing these tools for analyzing cancer genomes, they were growing their databases. They were taking, they were developing tools to do more sequencing, more whole exome sequencing instead of targeted sequencing of just a few specific genes. So early on, sort of the first pilot projects like glioma, what they were doing is they were just sort of collecting together certain particular genes they thought might be representative and looking at variation in these genes. But once they developed the tools to do more whole exome sequencing, they were finding a lot more mutations in these genomes than they expected. And so alarmingly, they showed that with larger sample sizes, the list of apparently significant cancer genes grew rapidly, and they thought implausibly. So it turns out that, and in some sense, this shouldn't have surprised them all that much, but a lot of the cancer scientists I spoke to spoke of this as a very surprising discovery, that there was a great deal more heterogeneity across and within cancer genomes than they had expected. So Tim Lay, when I spoke to him, said, yeah, we thought with the AML there might be 5 major mutations associated with it. But as we kept gathering more data, it looked like, hundreds of mutations were involved in driving these behaviors of these cancer cells. So in order to address that, they had to develop algorithms that ruled some things out and ruled other things in. Right? They had to have exclusion criteria to avoid these strong signals, right? That apparently looked like driver genes, but were instead just noise. And there's a variety, so the cancer scientists talk about heterogeneity in a heterogeneous way. That joke did not come out well, but so if there's heterogeneity across a patient of a cancer type, across different cancers, and then of mutation spectrum. So the types of mutations that you find within a cancer vary. So you might find more duplications, more inversions. And so all of these different kinds of heterogeneity led to all sorts of biases in the data and what they were gathering and what they were discovering as distinctive signals in these cancer types and subtypes. So they had to find ways to eliminate this noise. And they developed a variety of algorithms. The one that Lawrence et al. Speaks of in this, this paper that I'm citing here is MuteSig. And what they were doing is they were trying to avoid a version of what philosophers call the base rate fallacy, right? They were trying to eliminate variants that they were picking up that were just sort of part of the background noise. And so they used various tools to eliminate these variants. So for instance, if a gene is very large, there's a higher chance that you pick it up as a mutant, right? Because if it's going to be large, you're going to have more accidents along the way in replication, right? The timing of replication, so when it is replicated, at what stage of cell division, that affected which genes they were discovering. So there were a variety of different, signals that they were finding that were consistent across all these different cases, and they had to develop tools to eliminate apparent significance and rule these out in the process of identifying genes associated with this or that cancer type and subtype. Interestingly enough, now we're going back and revisiting a lot of this data from 2012, and it looks like, in fact, some of these genes that were eliminated might indeed have played an important role in cancer. So I think that there's a sort of interesting second wave of digging back into this data and questioning the very assumptions that were used to generate the consensus genomes in the first place. But that is just one source of error and bias in the genomic sequences that were developed as consensus sequences throughout the TCGA. So this comes from Mirzaman, who's a biostatistician at George Washington University. And what his lab does, they specialize in the problem of sources of bias and error in cancer genomics. And so there are many different sources. So all the way from the top to tumor purity and biosample quality. So it turns out that if samples are sitting in paraffin, they degrade over time. And when they degrade over time, you can get false signals. The purity of the sample, so when you take a biopsy, you don't always just get tumor cells, you get a combination of different cells, and you have to rule that out. And there are various tools for determining what percentage of these cells are likely cancer cells. There's differences in the process of DNA extraction and library prep for the technology process of like different sequencing contexts. And then there's just simply error rates in the instrumentation. So when we look at this genomic data, it is not a clean, pure source of unbiased information. There are sources of bias and error along the way, and there are various tools that have been developed to correct for these errors. And the tools are ideally, in principle, developed in service of addressing specific errors associated with specific types of cancer in specific places and times. But of course, one of the things that all the researchers said that I talked to is we're learning all the time. This is a process of ongoing correction. So it's not something that's been absolutely perfected at the end of the day. Another big issue that has arisen and people are paying a lot more attention to now is the fact that cancer itself is a dynamic process. So the samples that were taken for the Cancer Genome Atlas were samples at first diagnosis. But of course, cancer itself changes over time. Some cancers change more than others, and there can be heterogeneity in a population of cancer cells for a variety of reasons, either because there might be multiple different loci of origin, right, cells of origin, or because over time, as a cancer invades and metastasizes, it acquires variants that enable it in various ways to invade, to metastasize, to break down extracellular matrices. And the various events associated with these processes can be both genetic, epigenetic. They can be associated with tumor immune infiltrating cells. So there's a complex interaction between the tumor itself and its tissue microenvironment. And so an adequate picture of a cancer is not going to be based exclusively, of course, on its genomic variation, but also on all variety of downstream factors that play a role in how these genes get expressed and in combinations of various kinds. And then also, of course, the variation within and across a tumor over time. So for instance, in this image, you can see how DNA damaging treatment might lead to the evolution of novel variants that can resist various therapies, right, and grow out of a tumor that was initially treated successfully. And recurrence is unfortunately pretty common in a lot of cancer treatments, especially cancers that are discovered relatively later. So the next generation of cancer genomics or precision therapy research is of course is going to need to integrate all of these different sources of data, including but not limited to genomic data. So this is a paper growing out of that next generation effort. Mertens is trying to correlate not just the DNA changes, but also the downstream gene expression data in order to get a better picture of what features are associated with different breast cancers of different types and subtypes. So in order to understand how a cancer is likely to progress to respond to treatment, we need to know more about all of these features at all of these different scales, not only these genomic features. It's also becoming clearer that how deep we were sequencing. So the depth of sequencing has to do with how many replicates we're making. So if you do just 30x, you're looking at a certain limited number of replicants. That was sort of the standard for early on. Now it's starting to look like we need to sequence a lot deeper in order to get better, more accurate understandings of the variation within different cancers and across different cancers, but also taking samples across different parts of a tumor is going to yield potentially quite different genetic information. So this comes from a study that found that if you sample a tumor in different places at different depths, you can yield quite different results. And so with that new information, now people are going back into both the original samples in some cases and resequencing and reanalyzing this data, but also thinking more downstream that if we apply this data, we need to recognize that the variation is going to be much larger than we may have anticipated. So that's the upstream part. But the ambitions of precision oncology depend upon the quality and the validation of this data. But the genomics data assessment faces, I've argued, several methodological and practical challenges, both upstream with respect to sampling, with respect to recognition of the heterogeneity of cancers and their dynamics over time, with respect to how we're analyzing the data and what kinds of presuppositions we're using to exclude or include various information. With respect also, of course, to data storage and curation. So one of the big issues that people have been trying to manage is linking this genomic data with clinical data and making it accessible and basically readable, transferable across different contexts. And so different tags have often been assigned to different information, and making that uniform has been a big challenge. But then taking this downstream, integrating all this data in patients, so integrating clinical information about patients' sex and age and comorbidities and all these other factors outside of the genome, right, that play an important role at various temporal and spatial scales and how a cancer is likely to progress and respond to treatment, and integrating these extra genomic factors into our predictive models. And I read a fantastic paper that said we should stop talking about astronomical data. Now we have more genomical data. So we should all be talking about genomical amounts of data that we have to sort through. And this gets us down to this sort of transparency question. So in drug design and translation for precision oncology, there's something called the valley of death. Drug development in oncology is costly. There is a very high failure rate. And so one of the big challenges that people have been thinking about is how to sort of bridge this valley of death. On the one hand, you might say, well, okay, treating cancer is just plain hard. But there's also a serious issue here of maybe overextension or overhype of some of these drugs. And I'm not the first to suggest this. There are a variety of critics of precision oncology, and these are just a few. So Tanak and Hickman, Prasad has recently come out with a fantastic book. People have pointed out a variety of challenges and problems in the sort of downstream application of this data in clinical contexts. So Prasad et al. Have pointed out that the vast majority of cancer patients for the most part, don't seem to be benefiting terribly much from a lot of these new drugs, that they might have relatively limited or appear to have relatively limited scope of application, that a lot of the research is costly and perhaps poorly coordinated. So this comes from, this insight comes from a fantastic paper by Hay and a colleague where he pointed out that, for instance, there were 2 clinical trials looking at gene expression in ovarian cancer and developing targeted therapies for ovarian cancer, but different levels of gene expression were used as cutoffs. And so they found different results. The therapy was effective in one context and not in another, and people were sort of mystified why this would be the case, but just paying attention to gene expression level seems rather important. So that can lead to quite costly and unnecessary tests. Some of these biomarkers are improperly validated. So this would be another context in which we have to pay very close attention to gene expression in using these targeted therapies. A lot of the drugs, though there are some quite amazing successes, right? Recurrence, especially in sort of treatment-resistant cancers and end-stage disease, they can be, when you give them in combination, they can be quite toxic, but there's also a problem of recurrence. There's concern about testing against weak competitors. And then there's a huge literature on whether the endpoints that are largely used in a lot of these clinical trials, things like progression-free survival, are really good proxies for things that matter to patients, like overall survival. And last but not least, at least in the United States, some of these drugs are just prohibitively expensive. So there seems to be some issues downstream as well, facing extrapolation, unwarranted extrapolation, adequate collaboration and communication, shifting standards and cutoffs about what kinds of targets we're using. But then just this sort of basic issue of validating these biomarkers in the first place as good biomarkers for developing targeted therapies. And this, I recommend this book to everyone. It's fantastic. I would say it's based largely in the US. So one of the big issues that machine learning is supposed to solve is to reduce all of these dimensions. This is called the curse of dimensionality. In this post-genomic era, what we have is just this genomical amount of data. And the idea is that using machine learning, we can sort of sidestep this problem of this vast amount of data and reduce these dimensions and develop targeted therapies using machine learning that sort of identify the most important signal in all this noise that we've generated. But there's some real concern about whether using that data alone is going to be sufficient to address the kinds of problems that I've mentioned. So this comes from a fantastic paper by Carlisle et al., which is looking at attempts to extend the application of some of these very successful drugs in cancers that show similar features, right? And yet the long tail suggests that it's by and large not terribly effective. So I think this is just an example of the kind of concern that is being raised by folks like Prasad and Kimmelman and others that I cited earlier. This is a really expensive and costly process, costly in sort of time and efficiency, but also costly to patients. So if you do these 128 trials, right, 42 different disease interactions, 41 different drugs combinations, over 7,000 patients, and only 24% of the success. So I think the concern here is that we are thinking about things from the bottom up, thinking about things in terms of molecular biomarkers without attending to the variety, the diversity of cancer patients, and that can be quite concerning in terms of the sort of overall cost of these development of these drugs. So I would say there's 4 major challenges that are facing a lot of this drugs testing downstream concern. One is failing to attend to the complexity of interactions, and interactions in particular across different tissues and different types. So thinking about these targeted therapies as if the molecular information is the exclusive information we need in order to develop targeted therapies. Looking at endpoints that may or may not be patient relevant, concerns about success, in particular, the problems of recurrence, and just simple coordination of drug testing in effective ways, effective communication. So there are a variety of ways in which machine learning has been used to try to sort of bypass a variety of these challenges, sort of both at the upstream and the downstream level. And I've mentioned some of the upstream, but testing these drugs in cell lines is one way. Looking at images, as I mentioned, is another. And how successful has this been? So I'm going to look at some examples. So this was an effort to use machine learning in the context of screening mammography. And what they found, this was published, I believe, in 2020, is that radiologists outperformed the machines across the board. Maybe not surprisingly, but then also one radiologist in combination with machines did relatively well. So the intelligence of the machines did not surpass the radiologist's analyses, at least so far. Another application is using bioinformatics to identify expression patterns and identify risky biomarkers across different cancers to identify which ones are likely to be most aggressive. And so this was a study done by Hussain et al. Looking at expression patterns of various alleles associated with ovarian cancer. And what they found was that there were a set of genes largely associated with ovarian cancer. What's interesting, I mean, with aggressive forms of these ovarian cancers. But I've read a lot of these studies, but what's interesting about them, they're all sort of very similar because what they find is not surprising. So they find that there are genes strongly associated with aggressive forms of ovarian cancer or this or that other cancer that we knew were strongly associated with that ovarian cancer. And then they will find one or two gene or gene variants that are slightly above significance for that particular cancer, right? So oftentimes the question is, we're working with data and then we're discovering, it's unclear how much of this is a genuinely new discovery. There's also concern about how they're using cutoffs for significance. So it's, as far as I can tell, that sometimes it'll just say one standard deviation off from what is expected without a very clear analysis of what is expected and why it's expected. And then when you look at the algorithms that are used to develop these patterns of association, the theoretical background assumptions that are driving them are not clearly explained. And so it's unclear whether we can apply similar algorithms across different cancers. And then there's this concern about artifact. So a lot of the genes that we're discovering are playing significant roles in this or that cancer are genes that we knew in some cases decades ago from sort of old-fashioned wet lab cell and molecular biology were playing important roles in cancer. And so there's a concern about how much of this data is relying upon, how much of this research is relying upon data that we already have and bootstrapping that data to reinforce conclusions that we've already we've become very well aware of. So I think that there are sort of four major challenges facing these downstream applications of cancer research. First, the data they're working with is not theory-free. What we measure, how we analyze it, is shaped by a variety of assumptions and challenges and sources of bias we have to pay attention to. The interpretations of the data are not theory-free as well, of course. And cancer itself is a dynamic system. It is heterogeneous. And attending to the variety of different factors that play a role in each cancer's progression is quite important. Last but not least, of course, and this is the thing that matters, I think, most to patients, is it's not always clear what the scope of application of many of these targeted therapies is, and the study populations may or may not vary significantly from the populations we'll be applying these drugs in. And whether these surrogate markers are good targets for the things we're interested in is an open question. So I put this in just because I thought it was a fun new word. Apparently it is, integromics. So when I gave this talk to a lab that I work with at Washu, they said, well, what's the optimistic upshot? Here's my attempt at an optimistic upshot. So I think that it does seem that cancer scientists are paying closer attention to the variety of different scales at which we need to attend to factors above the genome. So I take it there are some sort of new efforts to integrate multi-scale data into our modeling and our predictive tools to develop new drugs and therapies, and I'm hoping that they're effective. But as I understand, these are sort of the hot new things, so we'll see how that goes. And that's it. Thank you very much, everyone.
Thank you, Anya. So if you wish to sort of react, you can use the reaction icons to clap, because of course we can't really clap all using the online system.
I never
so
found them. I never.
Okay, yeah. So it's at the bottom of the screen, and speaking about this, it's time for questions. Please click on participants, and on the right you can use the raise hand function. So if you have questions, please use the raise hand function. So Barbara, go ahead.
Okay, I mean, hi, Anya. Nice to see you again. Okay, no, I just a very simple question. It's just because you were speaking at the beginning about the base rate fallacy that these scientists were doing. Can you tell me more about this? I'm a little bit studying this in a totally formal way, but I would be happy to hear more.
So what I understand is the case is that if you assume that the same type and rate of mutations is the same across, I mean, this is an oversimplification, but if you assume the same type and rate of mutations is the same across every cancer, right, then you're not going to find, you're going to either find too many false positives or too many false negatives in your analysis. So adjusting for cancer type and extent of variation across these different cancers will presumably better identify, avoid these false signals, false positives and false negatives. And they're quite different numbers and types of mutations across these different cancers. So this image, it's quite small. I could blow it up, but it's called a gaddigram. Let's see if I can show you. Sorry. Oh, there it is. See at the top here, this is looking at the extent of heterogeneity of a specific type across different cancers. So perhaps not surprisingly, sort of childhood cancers, various leukemias are going to have a lower number and type than the say, smoking-induced lung cancers, for instance. And so adjusting for the extent of that variability, and there's different sources and types of heterogeneity, so you have to sort of adjust for all of them, is going to avoid these problems of false positives and false negatives. I'm explaining this at a very gross scale, but at the much more detailed scale, they have various tools for ruling out different kinds of error, like the size of a variant, of a gene, for instance, or the timing of replication.
Oh, that's extremely interesting. Yeah. I have another, I mean, this is really just, because I am now in a group of scientists, we are talking about testing for COVID and these kind of things. Do you think that the things that you're saying now also apply to virus testing? I mean, it's not your topic, so it's just a curiosity. Because there's a lot of difficulty in distinguishing pre-symptomatic from asymptomatic, which has a lot of policy implications, because if you can distinguish them, then you know that asymptomatic will always be asymptomatic, and then tendentially they have a different contagious rate from pre-symptomatic, that is people that then will get sick, for instance. And then I don't know whether you might have learned anything about this or came across anything in this direction. I mean, regarding COVID testing and.
I'm afraid I haven't looked at that, but that's fascinating. I'd like to learn more. I suspect that there's always problems with these base rate issues that come out in whether screening or analysis of large amounts of data like this with a heterogeneous sample. Yeah, absolutely.
This means, I mean, heterogeneity here is also related to simply social demographic factors in the sense of contagiousness. Because of course, if someone is just staying home, it contaminates people less. If you go if you drive by car, commuting, there is also this kind of things, not only genomic or other issues.
Oh, wow, that's really interesting. I wish we could sit down and have a coffee and talk about it.
Yeah, exactly. Me too. Okay.
So there's a question by Mary.
Hi, this is Mary talking here from Prague. I think that the presentation you gave is very interesting. I'm a cancer biologist, and to have an overview of all the Atlas Genome project and how it's created, to how we perform it and what we conclude, it's extremely interesting, but at the same time, to me, it's a bit frustrating, because when we started all this, our aim was to come up with a common factor across all cancers that probably would lead us to a cure. And when I see your summary today and all what we generated, I get a bit frustrated, because it's the conclusion that there is no common factor between all cancers, and we are not going to find this magic cure that we were hoping for. I don't know, maybe you could comment on
that. So let's see, you said that there was a hope that all cancers had the same common causal pathways associated with them, and that there would be one sort of targeted, effective tool that would allow us to treat all cancers, this sort of magic bullet, right?
Right. That would be our hope when we kind of started all this.
Well, I mean, as I understand, a lot of people are thinking about some of the new immune therapies as akin to a magic bullet, but I've read, I mean, you can find arguments on both sides of this issue, right? So this is exactly what happened when some of the earlier targeted therapies were developed. People got very excited that this is going to be the way to solve cancer. And I suspect that just the recognition of the variety, the heterogeneity of cancers has sort of keeps pushing back at that hope that there are some cancers that might be better controlled by immune therapies than others. There's some cancers that have apparently, the immune system can play both a positive and negative role. So interacting with the immune system can have paradoxical effects. And so it really, I mean, I think that each cancer and the tissue microenvironment of each cancer is likely going to be playing an important role in developing targeted therapies. And I imagine that is more like the consensus view now, but I don't want to speak for the cancer scientists, so I should what do you think? Do you think we're going to find a?
I think that the origin of the cancer in all different types of disease is so variable. The range of mutations and combinations of those mutations is so extent that actually I don't think, and that's just my opinion, but I don't think we are gonna find this magic bullet that it works, one works for all. So I still think that all the data we have been generating has been very helpful to understand the molecular pathways that are driving those disorders. But in terms of finding a cure that could fit all, that I think, and maybe I'm wrong, I really hope I'm wrong, but I think it's still very challenging.
Yeah. One cancer scientist I spoke, or he's a clinician and a cancer scientist I spoke to, I loved what he said, because he said, oh, all these scientists are saying we're going to, you know, we're going to do precision therapy. You know what precision therapy is? Chemotherapy. I give patients chemotherapy and it really works. He was talking about a particular chemotherapy for a particular cancer. And he said, this is 20 years old. We've been using it for 20 years. And of course, chemotherapy has lots of side effects, and there are a lot of problems with it. There are secondary cancers associated with it. And so I think one of the big motivations behind, you know, this whole project has been avoiding those terrible side effects associated with these drugs. But some of them are incredibly effective. There are these platinum therapies, for instance, can be quite effective for certain testicular cancers, and you know, there are unfortunately these drugs that are just horrible. They ruin your body in all sorts of other ways, but they do actually treat the disease. So, I mean, even if it's not a magic bullet, at least it's effective in some contexts.
John.
Hey, Anya.
Hi.
Good to see you.
Nice to see you.
Thanks for your talk. I learned a lot about some of the cutting edge science. It's pretty dizzying. My question is on this base rate issue again that Barbara brought up. The first thought that popped into my head when you were discussing that was, you know, didn't these researchers read Ioannidis' Why Most Research Findings Are False 15 years ago? I mean, but okay, but if you take this idea that we should expect a lot of false positives, you know, given that we're neglecting the base rate, one intuitive solution to this would be to inform the base rate, and you know, either, you know, if not do more Bayesian statistical analysis, then at least supplement whatever analysis we're doing with a richer understanding of the theoretical basis for the kind of associations we're seeking. And this would go to the kind of worries that a lot of people have, that a lot of this research is, if not you know, theory neutral, then certainly not as theoretically rich as a lot of other traditions in research. So I don't know if you have any thoughts about that, or if you know of any approaches that are more Bayesian and might include some, you know, biological plausibility in the actual statistical analysis.
Well, to be fair to the, I can send you the actually I'll send Barbara too, the Lawrence paper. What they do is I mean, you could formalize what they're doing as a kind of Bayesian analysis. So they go in and they say, like, these are the ones that we're finding that are mutated at much higher frequency than we would expect, like this olfactory gene. Why would this olfactory gene be mutated at higher rates? And so they found a very strong correlation between genes of a certain size, genes that were replicating at certain stages in cell division, between those and ones that were showing up again and again across these different cancers. And so in a way, they were updating their algorithm in light of this new information. And they really couldn't have known in some way that they would find these false signals until they did the sequencing and the analysis in the first place. So it was a very much, I mean, I think you could represent what they were trying to do over the course of these 10 years as a kind of Bayesian updating. But they really didn't know going in the extent or the variety of heterogeneity they would find. And so they couldn't have really done what they needed to do to rule out all those false positives. And I think, I mean, all of them said to me, look, we were just, when I said, well, how did you know how many samples to take of each cancer? Like, you don't even know how variable cancer is in the first place. Why would you know 500? And they said, we had no idea. Yeah, we had so much funding, and we tried to get 500 of each, you know, we did the best we could. So it really was a very much a bootstrapping effort. I don't know if that answers your question.
All right, so Barbara, go ahead.
Mine was a finger on the previous question, and it was just a curiosity as well. I mean, since all these big data, I mean, there is this issue with nutrition research. I mean, and there is Sana here who knows much better than me, because the inference there is even much more complicated maybe than this, and then pharmacology or these things like that, because there are a lot of dynamic aspects and then a lot of interaction. You have, I mean, in order to absorb iron, you have to take vitamin C or these kind of things, and then it interacts with lifestyle and this and this and that. Why don't we? I mean, I don't know. Maybe there is. Is there research that incorporates also nutrition in order to do precision prevention? I mean, not only therapeutics and pharmacology, but do prevention to prevent cancer. I mean, so I analyze the etiology of cancer and the prevention through nutrition or other things. Is there anything like that?
Absolutely. My sense is that, unfortunately, the vast amount of funding in the United States that goes to a lot of cancer research is looking at the downstream question, like the treatment, and not always as much prevention. And all the epidemiologists I've spoken to get sort of up in arms about this, that we're not paying as much attention to prevention as we should, both in terms of the research efforts and integrating it better with what we do know. But I take it that the All of Us project is attempting to do something like this. Though you should definitely ask Jim Tabery about this, because he has strong views about what all of us is really going to look like in terms of the output. But I guess the project is to integrate vast amounts of data, right? Not just genomic data, but also sort of patient patterns of, you know, exercise. I don't know how much attention they're paying to nutrition, but I suppose everyone's supposed to have a cell phone and be documenting what they eat or what exercise they get, what kinds of exposures they've had in various parts of the country. And
yeah, I think if there is anything really good about having these big data, is exactly to exploit them for prevention, since it would be a pity not to do that.
no.
So I don't see any hands raised, but I have a question myself. So you mentioned that the sort of radiologist outperformed the machine learning software, right? So how specific is this? Because I'm asking because of the reason that I've seen a presentation about sort of machine learning diagnosis and all that, and if you feed, for instance, in the cases of melanoma, if you feed, you know, pictures of skin cancer, the machine actually outperforms a clinician, basically. So there seems to be some cases where the machine learning is more successful than the clinical practice. But I assume that in other cases, it's still not as good as you know, promised.
Yeah, I mean, I can't speak for like a sort of, I haven't done a survey of the entire literature of machine learning on diagnostic, but I know that people at Washu here are actually doing research and collecting as many images as they can, and sort of doing retrospective teaching machines to learn based on this imagery. And I take it that it's, in some cases, it's in much earlier stages than others. And I would hope that as they get more images and more, they teach the machines better. But then when I have to say, when I talk to pathologists about this, they do have a lot of concern about what, I mean, when you've been doing pathology for 20 years, I take it that it's I mean, there's some, who's Polanyi? Is it Polanyi? That there's a certain kind of know-how that you acquire that is real, that I can imagine would be really difficult to teach. There might be some sort of subtle ways of which you know, it's really difficult to process this information. I don't know, but I mean, I would hope that they work in consultation practicing pathologists who really know their business.
Right. Well, the problem could be also with the differences in like how exactly the AI learns. You know, it could be on the basis of completely different criteria, as is the case with in other other instances. So then the sort of clinical or the criteria used by a pathologist could be completely different from the algorithm, and you don't even you don't necessarily know if you just feed in the picture sort of and let it learn, so to speak, how to recognize this. So I see another hand raised, but unfortunately we are out of time. So thank you, Anya, again.